Okay, I think we are all connected to YouTube. Good afternoon, everyone. I welcome all the viewers to the fourth Vyakhyaan in the series of 75 online or lectures being organized by the Physical Research Laboratory. As you are aware, we are celebrating the 75th anniversary or the Platinum Jubilee of the foundation of PRL this year. And to mark this occasion, we are organizing PRL Ka Amrut Vyakhyaan series, which commenced from 4th of August this year. The Vyakhyaan is scheduled every Wednesday at 4 p.m. unless it is notified otherwise. In this Vyakhyaan series or Vyakhyaan Mala, we invite eminent speakers in different fields of science, engineering, arts, and literature, not only from India but also from abroad. On behalf of the Director PRL and my colleagues, and also on behalf of the Amrit Vyakhyaan Committee of PRL, I extend a very warm welcome to Professor Chandrima Shaha, the esteemed speaker of today's Vyakhyaan, who is the first woman president of the Indian National Science Academy or INSA. We are very honored and privileged that she will be delivering today's lecture on a topic that is more relevant today than ever before, and that is our immunity and emerging diseases. Thanks, Professor Shaha, for gracing our celebrations of PRL at 75. I would also heartily welcome all PRL INSA fellows, invited INSA fellows, and the medical officers of PRL who have connected with us today live on WebEx for this Vyakhyaan. It's my honor to invite Dr. Anil Bhadwaj, Director PRL, to formally introduce Professor Chandrima Shaha and to invite her to deliver the fourth Vyakhyaan in the Amrit Vyakhyaan Mala. Professor Bhadwaj, please.
Thank you, Dr. Nanita. Namaskar. A very good afternoon. Vanakkam, Abhinandan, Kemcho. It's my privilege and honor to introduce today's Vyakhyaan Karta, Dr. Chandrima Shah. Dr. Shah is the president of Indian National Science Academy, in short, INSA. She is the former director and professor of eminence of National Institute of Immunology at New Delhi. Dr. Shah graduated from University of Calcutta and completed her doctoral research in 1980 from Indian Institute of Chemical Biology and holds a PhD degree from University of Calcutta. For her postdoctoral fellowship, she was at University of Kansas Medical Center from 1980 to 82, and then at Population Council, New York City during 1983 to 84. She joined the National Institute of Immunology at New Delhi in 1984 as a scientist. She has a long list of awards and honors which she has received in recognition of her achievements. And just to name a few, the J.C. Bose National Fellowship, the Ranbaxy Award of Basic Sciences, the INSA Shanti Swarup Bhatnagar Medal, Darshan Ranganathan Memorial Award, and the Om Prakash Bhasin. She is the elected fellow of all the 3 science academies as well as of the World Academy of Sciences. She has served as a council member of all the 3 science academies and was the vice president of INSA during the year 2016 to 2020. She is a member of task forces for several national and international committees. Her research has contributed significantly towards the understanding of cellular defense mechanism and modalities of cell death in multicellular and unicellular model organisms. She is a biologist by training, but is truly a multifaceted person. Apart from doing science, she has many hobbies, and which may now be a part and parcel of her life. She is a cricket player. In fact, the first vice captain of the cricket team from West Bengal. Further, she is the first female cricket commentator for All India Radio. And adding further to her cap of first, she is the first woman president of India, of INSA, in its 85 years of history. So she has a lot of first in her careers, and we are really delighted to have her delivering the PRL Amrut Vyakhyaan today. So ladies and gentlemen, those present live on this panel and those who have joined us live on YouTube, please join me in welcoming an eminent scientist of the country and President of INSA, Dr. Chandrima Shaha, to deliver her PRL का अमृत व्याख्यान on the topic immunity and emerging disease. Thank you, धन्यवाद. And now I request Dr. Shah to deliver her व्याख्यान.
Thank you very much, Dr. Bhadwaj. Now I would like to first I share my slides and then I go ahead with my. Is it visible?
Yes, it is.
So if I make it full screen. Okay, before I start, I must express that I'm very happy to be at PRL, albeit it's virtual interaction here, and it's distinguished. The distinguished guests who are today, the INSA fellows, the PRL faculty, students, and friends. So again, I would like to reiterate that it's a great pleasure, but these days you cannot, I mean, since you cannot visit each other, it has to be online, and it's a new mode of our communication. And I hope that this will materialize sometime in Dr. Bhadwaj coming to INSA, and I'm going to PRL one day. So even at INSA now, you know, the INSA building looks that nobody's coming these days, and so it's quite desolate. So I'm hoping that things will improve. So today I decided to talk about a little bit about the emerging diseases and the coronavirus. And it is almost now 2 years that we have been combating this little minuscule virus, which has really changed the course of our lives in a way. And it is our time to introspect about what we went through and improve our knowledge about the virus. And you know, we know a lot about the immunology of how we respond to viruses. But unfortunately, what we have come to realize that although we understand a lot of immunology, we do not actually understand the effect of the virus on the physiology, on human physiology. Our understanding is incomplete. So there is a need for more research, but there has been amazing progress in the last 100 years or so of immunology. So pathogens, you know, have been there since life evolved on Earth. And humans have been facing pathogens when they evolved from apes to the modern human. And so the virus, the bacteria, the fungi have co-evolved with the humans. And since they have a very shorter lifespan, they get more opportunity to modulate themselves. And that is why we have such a formidable array of pathogens that surround us. And we have a lesser time because our reproductive life is much larger than them. So this is a constant fight that we are going through. So during human evolution, as the different species of humans evolved, there was a fight between the host humans and the pathogens, and the evolutionary forces, the selective forces, were working on the host to prevent the pathogen and on the pathogen to create a successful infection. So this constant fight has actually given a very complex immune system to us, and to the pathogen many arsenals to actually counter the human the defense mechanisms of the human hosts. When humans were solitary creatures and had small families, the disease was not spreading that much. But as agriculture came, domestication of animals came, people started interacting more. And that is when the diseases started to spread. And slowly the epidemics came, then the pandemics came. And as you can see, the world population also increased. And there was a huge burst of human increase in the human population. So slides are not, yeah. So if we look at pandemics in the history, what we see, I mean, this is just to emphasize that how we have dealt with pandemics in the past. So we had in the 8th century, the Japanese smallpox epidemic. Then we had the smallpox came back again in the 16th century and killed about 56 million people. But the smallpox was dealt very effectively by vaccines and rampant vaccination and efficient vaccination drove the disease out of the world, and we are now smallpox free. So that was the main reason why smallpox could be eliminated, was because we had a very efficient vaccination program going on, and at that time also the population was much less. But then till 1980, when population is even bigger, the very efficient vaccination system worked. And another reason why the smallpox elimination worked is that smallpox did not have an animal reservoir. So most of the other diseases, they have an animal reservoir, and that is why they can jump from species to species and is sustained in the around the world. They're sustained around the world in various animals and even invertebrates, so they can be sustained like that. So the Spanish flu, which happened in 1918, between 1919, it was an influenza pandemic that was caused by the H1N1 virus. And there also the death toll was about 40 to 50 million people. And you know the plague, the Black Death, which actually wiped out a substantial amount of population in Europe. That happened in the 14th century. It came back again in the 19th century. And this is caused by a bacteria called the Yersinia pestis. But because plague sort of confined itself to endemic areas, a successful vaccine was not developed. There are vaccines to plague, but since we hear about plague very less, so that's why such a huge program like a smallpox vaccination was never initiated. Coming to more recent times, we see the HIV. Now it's been 40 years and we have not been able to really formulate a vaccine against the HIV virus. So it's been very difficult. And SARS vaccine which happened in 2002 and between 2002 and 3, that vaccine is still in development, and it was discontinued because SARS infections went down. There is no vaccine for MERS, which is Middle East respiratory virus. There is no vaccine for that. And Ebola virus is a disease which is a deadly disease, which happens mostly in Africa. There is one vaccine which is there, and Ebola has somehow the spread of Ebola has been contained. Otherwise, Ebola virus is a very dangerous virus. And there are several vaccines now for the coronavirus that we know that these vaccines have been developed very fast indeed. Vaccines cannot be developed that fast, but due to emergency, due to evolved technologies, we have been able to develop the vaccines for corona. Now, there are about 212 million people infected and more than 4 million people died of this coronavirus. And we know many of our near and dear ones have suffered from this disease. So this is something that we are currently facing. And every day there is like a new data, new realization is coming in encountering this virus. Something like this never happened in human history. Now, if you look at the smaller epidemics that has happened between, I think it's 2011 and 2017, you can see that, diseases like cholera, Zika virus, meningitis, shigellosis, and all these that you read, they have come in smaller form of epidemics and has really killed people, but not to that extent. This is because of better medical care and also because it was not allowed to spread. So, but however, we have these diseases. So you can imagine how many diseases we are dealing with currently. Yeah. Now, this is a chart that has been developed by Anthony Fauci of NIH. And this is about the emerging and re-emerging infectious diseases. Now, the brown labels you see are newly emerging diseases. And this is a chart of 2017. I do not have access to his latest chart. And re-emerging diseases are the blue ones. And the black is the deliberately emerging, which they have labeled anthrax. Anthrax they have labeled as deliberately emerging. So you can see how many diseases we are exposed to and how many, probably now the chart is much bigger and we have all these variants and the coronaviruses. So this is the scenario. So it is a difficult scenario. So we have to really work hard on discovering these and anticipate the occurrence of these diseases causing, these viruses or bacteria causing the pandemics. So what are re-emerging diseases really? So it happens like previously unknown infectious agents that are around that we have never found them are emerging. So these previously unknown agents are causing many diseases. Known agents that have spread to new geographic locations or new populations. So known agents in one part of the world goes to another part of the world and spreads to a new region. So that is also there. And then known agents whose role in specific diseases has previously gone unrecognized, that we have not been able to recognize those pathogens. They cause a lot of diseases in various parts of the world. And then re-emergence of pathogens whose incidence had significantly declined in the past, but whose incidence of disease has reappeared. Now, this maybe the re-emergence of some of the pathogens are due to reduced vaccination. I will show you in some cases. So, but those are known pathogens. And also evolution of pathogens over time. So a given pathogen evolves over time. And as I said, this constant war between the humans and the pathogens, and also their passage through animals, are causing new and new pathogens to evolve. So in immunology, the father of, if we call father of immunology, it's Elie Mechnikov, who was a Russian scientist. And he was the one who first showed that if you take a starfish larvae and put a rose thorn in it, the cells sort of home around the thorn as if to throw it out. So this was the first description of an immune reaction by Mechnikov. And he was like a polymath, and he was awarded the Nobel Prize in 1908. He did a lot of work on how pathogens are engulfed by human cells and what the human cells do with it. So he actually, since his time, immunology has really developed. So in very short, if I can give you a bird's eye view of immunity, we have two kinds of immunity, the innate immunity and the adaptive immunity. The innate immunity is where dendritic cells and macrophages are there. These are two types of cells, immune cells. There is also neutrophils, but these two cells are very important because they first encounter the pathogen. They actually engulf the pathogen, chew them up, and express their antigens on their surface so that if and when required, this happens within few hours, if and when required, the T helper cells actually recognize these antigens and tell the B cells that this antigen is here. So either through memory response, if it's a second infection, or through a response to this particular antigen, the B lymphocytes will generate antibodies. And the helper T lymphocyte may also can initiate cytotoxic T lymphocytes. So this cell-mediated immunity actually can kill the incoming viruses. So this is in very short is the immune system. The immune system is highly complicated, but I'm giving you a picture which is actually a schematic. So this adaptive immunity comes in days after the innate immunity kicks in. Sometimes innate immunity is sufficient to get rid of the pathogen, but later on, adaptive immunity takes over in case the innate immune response is not efficient, and then the pathogens are dealt with. But this is also the principle of vaccination, as I will show you later. So as this is a macrophage, a beautiful picture where the macrophages are taking up the streptococcus bacteria, and what they do with any pathogen, they actually put them in a cellular sac called the phagosome, and the phagosome fuses with cells phagolysosome. With cells lysosome, lysosome contains the lytic enzymes which digests material, and lysosomes are normal part of our cells, and they are used to recycle materials. So the phagolysosome that is formed, they are chewed up, the pathogens are chewed up, and the two important things happen to the pathogen debris. Some debris is gotten rid of because they are not essential, and maybe they recirculated back the raw materials, and many of these antigens are displayed on the surface of the cells so that the T cells can come and recognize these antigens and tell the B cells to generate antibodies, or tell the cytotoxic T cells to kill the pathogens. So this is in a nutshell what happens when a pathogen comes in. Now there is many years ago we showed that actually within the phagolysosomes, when these dyes, sophisticated dyes became available, that they actually generate reactive oxygen species, and how they kill these pathogens are that the NADPH oxidase, which is on the phagolysosomal membrane, will convert oxygen to superoxide, and superoxide will dismutate into hydrogen peroxide, and hydrogen peroxide then generates very reactive radicals like the hydroxyl radical, like the peroxyl radical, and in the presence of nitric oxide, the peroxynitride. And these are sufficient to kill the pathogens. Sometimes, if we get disease, that means our systems are not working properly, and maybe this system also is not working. So the other cell that I talked about from the macrophage is the dendritic cell. So the dendritic cell also presents the antigens to the T cell. This is again a pseudo-colored scanning electron micrograph, but it is really looking at life up close. But if you look at the macromolecular details, you will see that the dendritic cells are actually talking to T cells through multiple molecules, which are cell surface molecules. And this is the little antigen that it is trying to present to the T cell so that it can tell the B cell to create to generate the antibodies and also to the cytotoxic T cells. So these are MHC molecules which are on the dendritic cells, and they carry the antigen. And these are co-stimulatory molecules that are present. So this interaction is actually a very intricate and complex interaction. And in case of immune insufficiency, one of these may not work. So in the adaptive immune response, again, you see this is a macro. This is a very beautiful picture to me. This is a macrophage. Macrophages usually grow in culture as flat cells, and the macrophage and a T cell is sitting here. And what happens is that the streptococcus, it will pick up the streptococcus, it will present the antigen through digestion to this T cell, and this T cell will tell the B cell to generate the antibodies. So this is the principle of vaccination. So the vaccination will generate antibodies against a dead or inactivated pathogen or pathogen proteins that are injected in your body through either directly protein or through mRNA or through DNA. So these vaccines generate through this principle that whichever way the protein is dealt with by T cells, B cells, and then the antibodies are generated. So this is how the vaccination works. Now, the ideal vaccine not only protects one from disease, but actually ensures that one is not a courier. And if a sufficient amount of people are vaccinated within a population, then we sort of get herd immunity. It has been very difficult to get herd immunity in India because our population is very large. And at a given place, if you have sufficient number of people vaccinated, then the disease will not pass to the non-immune population. So just to show how vaccinations over eons have been actually very effective, if you look at the measles vaccine, these are the number of cases, 800,000 we used to get in 1980s, and by 2016 it was about 4,000. Now the places where measles cases, where measles immunization was going slack, the numbers started increasing. So directly it shows that vaccination is very, very important. And similarly, I mentioned smallpox. Smallpox was eliminated in the 1980s from the world because there was very efficient immunization program. And in these cases, you see the very large amount of incidences happening in these years. But smallpox was not only killing people, it was disfiguring people. So it was very important that smallpox was driven out of the world, and it did. I mean, this is a very rare example, but it happened that smallpox actually was eliminated. The polio cases in India, the polio cases went down, and through very active vaccination programs. So I showed these slides to assert the fact that vaccination is a way to go about, and vaccination is very important, not only for yourself, for the others of the society as well. Now we come to this by now familiar picture of this coronavirus, who is supposed to be carried by bats, and these viruses have really taken over our lives. And so these viruses, the original Wuhan strain, which was found in Wuhan, actually generated variants. So there is an alpha variant, there's a beta variant, there's a gamma variant, and delta variant. And each of them have many mutations, and the mutations makes it either very infectious or easy, so that easy transmissibility is there, makes it more virulent. So it's actually a virus will not like to kill its host, because then it is killing the reservoir in which it is growing. But this is happening, that people are actually dying because of consequences of the virus infections. But if you look at the amount of people that are dying versus what happened in older times, it is actually less. So the alpha variant occurred in Great Britain, and it spreads easier than the initial one variant. And then the beta variant occurred in South Africa. The first, it was detected in South Africa. And it has a high possibility of causing illness or death. Then the severity of the disease is much more in the delta, which was found in India, and much of our second wave was due to this delta variant. And the gamma variant was first detected in Brazil, and the strain is very contagious. So right now we have these variants which are going in, and they have mutations. And so the mutations occur. Mutations are very important for the virus, because that may render the virus ineffective or make it more effective. So in China, this variant actually gave this B1.1.7 and B1.351, and in the Brazil strain. But what you see here is that some of the amino acids here are very prone to mutations. So this 501 is very prone. And so what these mutations do are they cause more efficient transmission, or reduce antibody binding and immune protection, or reduce vaccine efficacy against all these strains. If you look at the coronavirus, actually, I think you're very familiar with this, but I'm just going through this, that there is a spike protein in it, and the RNA, it's an RNA virus, so RNA is within the virus, and this spike protein is like a trimer, and it has two parts. One is S1 and one is S2. S2 is the binding part, which latches onto the mammalian cells through ACE2 receptor, angiotensin converting enzyme receptor. It first latches it on, and then there is a protease cleavage, which allows this fusion peptide for the viral membrane to fuse with the mammalian cell membrane. So this is in a nutshell what happens with the virus. And so the spike proteins are very important. And what is interesting, very recent findings show that the spike proteins are extremely flexible. So they can bend. So when they're binding, they actually are very straight, like a very straightforward, and go and bind to the cells. But in other times, they can fold themselves if they want to avoid the immune cells or any or the antibodies. So this is a very smart thing that the virus does. So this is a schematic which shows that this trimer of the virus, the spike protein virus, it binds to the ACE2 receptor. And it's actually in one dimension, but if you can see here, these are the residues that are very important actually for binding to the ACE2. And the mutations, as shown here from a structural biologist viewpoint, that these amino acids which are at the binding site are the ones which undergo most mutation. And this gives the virus some advantages and may not give advantages as well. So, these are the amino acids that, and there is a deletion here. There is a deletion, there is a amino acid change, there is amino acid change here also. And that causes this to bind with more affinity, so that it makes it more contagious. So there is now this virus comes in and binds to the ACE2 receptor, but this is actually helped by a protease cleavage, which is, if the protease belongs to us, it is TMP RSS2, or it's furin, which is within our cells, and that helps the cleavage of the, as I showed, the cleavage of the trimer at some point, where the cleavage, they don't actually fall apart, but it exposes the sites which actually help the virus membrane to bind to the mammalian membrane. So these are the enzymes that are within us. And I'm sure during the course of evolution, the virus the selective pressure has forced the virus to use the cell's own enzymes to sort of use it and to use it to its advantage and create these infections. So the mutations can be, it can be highly deleterious, but they are rapidly purged because, as I said, the virus normally doesn't want to kill its host, because then it also goes away with the host. So that is the principle in which evolution works. So high effect mutations that contribute to the virus adaptation, that is why they tend to be less. And a small minority of mutations actually impact virus phenotype. So there are some mutations which confers a fitness advantage to the virus. And that fitness advantage is so important that these mutations are actually nurtured and kept. And mutations may alter virus biology, like pathogenicity, infectivity, transmissibility, and antigenicity. And while we are studying this, we have realized that although we know much more than we used to know, there are many aspects that we really don't understand. So we need more and more studies to understand the pathogenicity of the virus. It's very important. And the rate of evolution of the SARS-CoV-2 from actually December 19 to October 20 was consistent with the virus, approximately 2 mutations per month in the global population. But this is a slow rate actually, because it can be, there can be more mutations. I mean, there are viruses which have more mutations. So what we get is 2 mutations per month, which is also not easy to deal with, as I will show you that there was a mutation at amino acid 614. It was a aspartic acid residue in 614 of the viral spike protein. By April, a single mutation in the genome occurred, and the aspartic acid was converted to a glycine. Now, this variant is all and has replaced the original 614G variant, and this provides some sort of selective advantage to the virus. It transmits more easily than the other one, and the number of functional spikes on each viral particle is 5 times higher because of this mutation. And patients infected with this variant has higher viral loads in their upper airways. So the main area where the virus actually infects is the nose, nasal mucosa, and the upper airways. So that's where the load is higher with the 641G. And that leads naturally to increased transmission when one is coughing. There is another substitution which is of concern. It is called N439K. It changed the asparagine at the 439th site on the spike protein to be replaced by lysine. And this emerged and circulated widely in European countries and was the main cause of infection there. And it enhances the binding affinity for the ACE2 receptor. So naturally, if the virus has an increased binding affinity for the cellular receptor, the transmission, the efficiency of the infection would be more. And it has been dissected out, and what has been found is that the binding energy of the N439 complex was higher than the wild type complex. And the complex with the mutated virus forms, there are more hydrogen bonds than the wild type complex. So there is at the intricate at that site, there are more hydrogen bonds. And the stronger binding, it is predicted, could be due to replacement of the asparagine with lysine, and it forms a new salt bridge in the complex with human ACE2. And this could increase the electrostatic interactions. And the complexes are also bound by van der Waals interactions and polar solvation free energy. So these are very intricate changes that happens when a mutation takes place. And this N438 came, sorry, it's 439 actually, I've written 438. 439K mutation is actually reduces the neutralizing activity of the antibodies that are generated in our body. So even if you are vaccinated, you may get the infection, albeit in reduced strength, but you can get because this vaccine escape happens with these mutants. There is another very important substitution which I showed, it occurred in all the variants. That is the 501 mutation, where it is a substitution in one of 8 spike protein substitutions present in the UK variant, and has also appeared in convergently in, this is convergent evolution in a way, Brazil and South Africa variants. Now, it is a virus of concern because it increases transmission. What has happened here is that the asparagine has been converted to tyrosine at that level. And so there are very complicated interactions have happened. And if you see, the mutation has happened just at the site of the trimer, one of the trimer, this is one of the arms of the trimer, this is the another arm, and this is the another arm. So the mutations have happened at a point where it is binding to the ACE2 receptor, and that mutation has changed its affinity. So, so this is a very important mutation, this 501, and occurs in all of these variants that we see. So now Delta variant is the major virus of concern in India, and we are seeing Delta plus now. And as I said, that most of the second wave was due to the Delta variant. And the Indian SARS-CoV-2 Genomic Consortia collects a percentage of the coronavirus positive samples for analysis from all states. And there are about 50,000 samples analyzed for their genetic composition. About 2/3, that is 30,000 to 130 is the figure given, were among the internationally verified variant of concern or variant of interest. So there is a variant of concern and there is a variant of interest. The variant of interest can convert to variants of concern depending upon its ability to infect. So of this analysis, nearly 20,000 were Delta variant and about 5,000 its associated lineages. So rest of the 5,000 was other variants. So Delta is about 40 to 60% more transmissible than Alpha and almost twice as transmissible as the original one strain. So again, the airways of the patients get more virus with these mutations. They become they turn out to be having more affinity for these cells of the upper airways and the nasal mucosa. And in India, the new cases like Delta plus, like these are the AY1, 2, and 3 are the Delta plus, which has been found in Rajasthan and Maharashtra. And they are surging in, actually surging, Delta infections actually surging in highly vaccinated countries as well. So this is really a variant that, we really don't know how much vaccination helps against the Delta variant. But it has been maintained in the literature that even if you get sick with the Delta variant, if you're vaccinated properly, you will not get the disease to what it actually can cause in unvaccinated people. So to do that, we had these vaccines. I'm sure you know about these vaccines, but just to say that we have the killed vaccine, which is like for us, the Bhadwaj vaccine. For China, it's Sinovac and Sinopharm. They have also taken the virus and killed it. And this killing are by formaldehyde fixation. And these are given to you. And the macrophages, they recognize the antigens. So you do not have an infection because these are killed. And you get an immunity by the process that I told you for against the virus, against the SARS-CoV-2 virus. So in clinical trials there is another vaccine which is by Shifa Pharmed, is in Chinese Academy of Medical Sciences. They have in clinical trials. And there is an attenuated vaccine, but attenuated vaccine, people are not very happy to actually do it because the risk of attenuated vaccines is more because the virus are not killed and they are just sort of inactivated a bit. But if some live virus remains, then or some virus gets activated, then of course immunocompromised people will have a lot of problems, as well as the people who has not received any vaccine before. So one of the example of attenuated vaccine is the polio vaccine. There have been cases in which it has caused polio, but very, very less, in very, very less quantity. And in clinical trial, one attenuated vaccine is there, Codagenics. For the generic vaccines, we have where the RNA of the virus is that by not exactly the whole RNA, the spike protein RNA is actually given inside liposomal particles and injected. And these are the Pfizer and Moderna vaccines, and that generates protein in your body using your cell's own machinery, and that those proteins are presented to the T cells. The T cells will talk to the B cells, and against those proteins, you will have antibodies. So in that way, Pfizer and Moderna vaccine has been very successful, and they have given protection up to 98%. So in clinical trials, there are other vaccines of RNA, these RNA vaccines, CureVac and Inovio Pharmaceuticals. So, and the other are viral vector vaccines, where one uses a viral vector, which is like an adenovirus, which causes common cold, and the spike protein DNA is put in those vectors, and the vectors are injected in the human body. The vectors will go to the cells, and they will inject the material inside the cells. Again, the cellular machinery will make the proteins. The protein will be expressed on the surface, and the T cells will recognize, and macrophages will pick them up, and T cells will recognize, and B cells will generate the antibody. So that portion is similar. So we have an example in Ebola vaccine, and the other one is the AstraZeneca vaccine, where they've used a chimpanzee adenovirus, and it is also one of the successful vaccines have been recommended all over the world, and which is our Covishield vaccine actually. So the Janssen, CanSino, and Gamaleya is the Sputnik vaccine, which is also similarly made, and it is approved in India now. So what about the vaccines and variants? So there has been a study that has been done where about 300,000 randomly selected people across the United Kingdom has been taken, and they have been surveyed from December 2020 until 16th May, when Alpha variant dominated, and 17th May to 1st August when Delta was dominant. And protection against symptomatic infection actually decreased significantly for the Delta period. So Pfizer, which was 98% protective, became 84% protective. And AstraZeneca, or the Covishield, it's not done with Covishield, but AstraZeneca vaccine also, from 97, 98% to 71%, its efficacy decreased. So the efficacy of the 3 US authorized vaccines also dropped, and these are all due to because the variants are happening. Dropped from 91 to 79% between May and July as Delta took over the region. So these variants are causing these problems with the vaccines. And but that is why the talk of a cocktail vaccine with many of the areas of the RNA that in the spike protein. So it's a cocktail kind of thing. Instead of 5, 6, 8, we actually don't know exactly what epitopes that the companies have used. It's not made public. So so many more epitopes should be given so that the cocktail vaccine actually gives you an opportunity to respond when you are infected with the variants, whether it's the Delta variant or it's the Alpha variant, and with their own mutations. So because of, for people, it's particularly difficult for people with weakened immune systems. And whether because of age or disease, boosters can offer important protection. But see, when we have taken a vaccine, suppose I have taken a vaccine in March, and it's already about 4 or 5 months, and how long the vaccine immunity stays? They're saying that it's from 6 months to 9 months. So after that, ideally one would need a booster. But given the situation that vaccines are not available that much, I don't think we'll be getting the booster. The third booster we will not be getting soon. But it is necessary to have a third booster because then you are immunized. Otherwise, you go back to sort of ground zero again, and the herd immunity is not achieved. So if we look at the example of Israel is a very small country, and it's about 78% of the population is immunized. Now in Israel, the recurrences of infection is occurring because it has one of the world's highest infection rates. Because although they are all vaccinated, I mean, most of them are vaccinated. The question is now whether the current surge is due to waning immunity that I was saying just now, or because the Delta variant is more robust and it is able to infect humans more than other strains. I think it's a bit of both, because if you look at this graph, so the first vaccination happened in 19th of December, they started vaccinating. Then the incidences went down. Then here, the Delta variant was identified. Again, the incidences are going up. So Israel is like a natural laboratory that one can do these studies and see that how immunization has actually helped or did not help. So what is the future? The future is that, we have discovered much about the SARS biology, and all said and done, and extraordinary strides have been made with COVID-19 vaccine in a very short time, and a lot of data on what is the biology of the virus, what are the physiological consequences. Although data are incomplete, we have not had enough time to look at the data because the time to have the data, because the timing is very short. So for this, the funding needs to be improved, because we really need to look at other components which are helping the immune system to work, and to see if we can reinforce those components. Remember when I showed you that when ACE2 is actually binding to the spike protein, there could be soluble ACE2 be given so that the receptors are preoccupied. And some of the pharmaceutical companies are working on those kind of soluble agents. And the funding really needs to be improved for basic research on this, because not everything is applied. And the effects of virus on the human physiology studies are also very important. And a lot of it happened because this was a very highly infectious disease. So the autopsies and studies that we normally get through pathological observations, we could not get. So the future will also depend on the ongoing evolution of the SARS-CoV-2. It is impossible to predict how the virus will mutate and how it will behave. But at the same time, the behavior of the people and the government's decision to implement whether lockdown or use of masks or use of hand wash is very important, because it has happened that use of masks has been discontinued in some of the countries, and they are getting increased infections. At the high level of vaccine coverage, as I said, it's us being a very huge country, it is difficult to get because not so much vaccine is available. But I think the vaccine hesitancy is also another thing which is contributing to this. People should be taking vaccines on time. And this high coverage has to be achieved, because the premature premature stoppage of mask wearing, as I said, and social distancing could be catastrophic. And we have been seeing this cricket matches, this football matches where people are sitting without masks and actually shouting, and the virus, if at all with them, is going out in the atmosphere and to your neighbor and all. So these are super spreader events that has happened. So one has to be concerned about this, because today in my country it can happen, but tomorrow it will affect another country, because this pandemic is actually all over the world in almost all countries. So so one has to really country-wise, one has to really be very strict, but different governments have different ideas. So it's very difficult to say that when we will actually get rid of this virus. So we have learned so much, but yet we have so much left to learn. So I just gave you a bird's eye view of what the situation is right now with the coronavirus. Thank you very much.
Thank you, Professor Shaha, for a very insightful and very stimulating Vyakhyaan, and discussing in depth in general about the complex life and evolution of viruses, and especially discussing about the deadly virus that which we are dealing with currently. I'm sure everyone would agree with me, and I'm also sure that many of you are anxiously waiting to ask questions to Professor Shaha. So I now invite Dr. Lokesh Sahu and Dr. Kuljit Marhas to conduct the question and answer session. Over to you, Dr. Lokesh.
Yeah, thank you, Professor Srivastava, and thank you, Madam, for very excellent talk. So we will be reading the questions which is posted in YouTube chat box first, and then we will take questions from the WebEx, the audiences who have done it through the WebEx. So the question I read is: humans gain permanent immunity to the highly contagious chickenpox virus once they are vaccinated or get the disease once. This does not happen in COVID, for example. Is it known why this happens?
No, it is. It is actually not known why this happens with the chickenpox virus, and there are other one or two examples where you get a lifelong immunity. So this is actually a mystery of immunology. Is why viruses like COVID, the SARS, other coronaviruses that we have, that we do not get a lifelong immunity. But there are several predictions on this, that could be that the memory B cells that are formed in response to a chickenpox infection or a chickenpox vaccination are very sturdy, and they are kept in the lymph nodes. And whenever there is an infection, if there is an infection, then they are called from there, and after undergoing the very strict checking, they do come in the system, and they act as generate antibodies or generate cell mediated immunity. So it is because of the kind of memory response it has. So for the coronavirus, there is not that kind of immune response that will sustain, because even if you are seeing with vaccination, the antibodies are not staying beyond about few months.
So I read another question, which is during the height of the COVID second wave, the term cytostorm was used extensively as something that triggered organ failure or death. What is cytostorm?
Cytostorm is actually an immune reaction. So where, because the body sometimes, these T cells, particularly the T cells, it gets sort of confused and starts generating a lot of cytokines. It secretes these cytokines, which are actually pro-inflammatory. So if pro-inflammatory cytokines are more in your system, then you get a lot of inflammation in the organs, and eventually it leads to organ failure. So the cytokine storm that was happening, and that's why they were using steroids to sort of bring down the cytokine storm. But the important thing was the T cells getting overactive and secreting a lot of cytokines. So this, why it happens, we don't know, but it does happen with some people.
So another question is, why vaccine for HIV is still elusive?
It's difficult to say. It's about 40 years that we have been working on this HIV vaccine. And, you know, for some reason, this HIV vaccine has actually failed. That's what, at the initiation of the COVID vaccine, it was a real worry that we will not be able to generate a vaccine for COVID, for SARS-CoV-2. But for HIV virus, somehow we are not getting a response that we should. I mean, there are candidates, but they are not very effective.
Yeah. So another question is, does intestinal epithelial cell, that means IEC, has any role to fight SARS-CoV-2?
Intestinal epithelial cells.
Yes.
So intestinal epithelial cells do get, I mean, they actually take up SARS-CoV-2, but whether the immunity, what happens there, we really don't know, but it is suspected that in the intestine, our microbiome in the intestine is involved with response to SARS-CoV-2. It has not been proven yet, but as like in many diseases, our gut microbiome, which is actually the beneficial bacteria that we have, helps us to have better immunity. So it could be something like that. So we do not have a complete knowledge on what particularly intestinal epithelial cells do.
Yeah, so I'll go through another question. That is, there was some news for the mixed application of Covishield and Covaxin. How much is it useful?
There was an idea. See, ideally, if you have taken Covaxin first, so you've been exposed to many epitopes of the virus, because that's a killed virus vaccine. So when you take Covishield after that, which has about 8 or 9, I think, spike proteins, so you'll get a booster for antibodies to those particular spike proteins if it were exposed epitopes in the Covaxin. So we are not sure if that is happening, because we don't know what are the epitopes that are there in the AstraZeneca vaccine. When it was said that you can take Covaxin followed by Covishield, Dr. Soumya Swaminathan and WHO actually said you should not be taking it. So there is a confusion as to whether you can take the two vaccines. But ideally, if you think about it, if you think about the theory of it, the Covaxin, which has many epitopes, actually can the Covishield can actually create more antibodies for those particular epitopes that are Covishield, that are being exposed by Covishield.
Yeah, thank you. Another question is, what could be the reason of present spatial heterogeneity in coronavirus cases in India?
Pardon? I didn't get that question.
Yeah, so the question is, what could be the reason of present spatial heterogeneity? Spatial heterogeneity.
Heterogeneity. I think
cases in India.
We do not have enough data on that actually to make any conclusion. So the spatial heterogeneity in the cases that are happening, we have to see a larger number of cases to make any conclusion on that.
Okay. So another question is, does probiotics offer any hope, or there any evidence for that?
As I said, that the gut microbiome in other diseases, the gut microbiome has been shown to be protective. So probiotics is a form, when you take probiotics, like, you get those in the markets and also in tablets. When you take probiotics, what you're actually doing is replenishing your useful gut bacteria. So you are fortifying your gut bacteria in a way. So not all of us are in the best of health all time. So if you take that, it might help in general your immunity, and that can help with the fighting the diseases, including the corona.
Okay. Thank you. Another question is, there are different scientific beliefs for COVID vaccination. One school believes no vaccination is needed once the person got infected. Second believes at least one dose of vaccine is needed for those who have had COVID attack and subsequently recover. And third is believes full immunization. Your comment, please.
Yes, see, the belief that no vaccine is needed once you have got infected is based on the premise that an antibody has been generated. It has been shown that if you are very ill, if you have a vigorous disease, then you get more antibodies. The antibody response is more vigorous, and that may stay till a year or 9 months or 9 months. And if you have a lesser disease, the antibody may stay till 6 months. Now, it is not true that you don't need a vaccination. Once your antibody wanes, you may need a shot that could act as a booster for the antibodies that you've had. So it is important that you take a vaccine. Since we have, as I kept on telling, that we haven't got enough time to see what is happening. So 6 months, 3 months, 1 year, we have no idea. But say if you take a view of say 9 months, after your disease, you should take a booster vaccine, because it is not everlasting. These antibodies do not last long.
Okay. So another question is that how many epitopes are generally considered for antibody cocktail?
The pharma companies, they don't tell how many epitopes they are actually using for generating the vaccines. I would suspect that there are 8 or 9 epitopes. They are targeting the very specific amino acids in the region of the binding of the virus spike protein to the ACE2 receptors. And that against those they are generating. So would be 8 or 9. I have no idea actually.
Yeah. So another question is, can someone have another type of vaccine after 6 months of having both doses? Say earlier Covaxin both doses is taken, and now Johnson single dose can be taken after 6 months?
Theoretically, one may take it, but science is based on evidence. The time has been so short that we do not know if people have taken 2 vaccines and then they have taken a third vaccine, so what has happened to them. So that data we do not have, and we do not have the immune profile of such cases. So I will not be able to comment on that.
Another question is, what are the critical side effects reported in India due to COVID-19 vaccination?
Side effects in that sense, in England, the AstraZeneca vaccine had a blood clotting problem, but I have not read any reports in India about the blood clotting problem happening with the Covishield. So it also depends on the ethnicity where the vaccine is being given. AstraZeneca was actually stopped in between because of those clotting problems. But in India so far, apart from fever and for a few days, I don't think there is any serious problem, and even if it is, it's in very, very small quantity.
Yeah. So still we are getting a lot of questions. Your talk has been very exciting. So I'll go through another question. In the plot showing global reemergence of infectious disease, why there is a lot of scatter in US?
US. So
yeah.
US also has a lot of infectious disease coming up. So that is the scatter. That is why the scatter is there. The scatter is all over the world actually, if you look at it. But in the US, there are diseases that are coming up. Even the tropical neglected diseases are coming up in the US. So because of the travel, a lot of diseases are exchanged between the countries.
Okay, Professor Bhadwaj has a question.
Can we? So in between, so we have some more YouTube questions, and then we'll take away from WebEx. So anyway, so before going to YouTube, I'll request our director, Professor Bhadwaj, to ask questions.
Yeah.
Thank you, Lokesh, and
Yes.
thanks to Professor Chandrima Shaha for a very excellent and highly informative Vyakhyaan. I understand now that virus needs a host to survive. And in between, we are talking about airborne virus. So my first question is, how long will this virus, coronavirus, survive in the air before it's going into a host body to really get survive?
So in the aerosol, when it is in the aerosol, it's being carried. See, the heavier droplets, if you cough, heavier droplets, they fall faster, and so they are not of concern. But the finer aerosol drops, they can remain suspended in the air according to literature at about 2 to 3 hours. They can stay. But my suspicion is that if you have coughed and then there is free flow of air, then it dilutes. But if you are in an air conditioned room, then if the suspended particles are there for 2 to 3 hours, then the surrounding people are liable to get infected.
Okay, you also talked about mutation, and you talked about alpha, beta, gamma, and delta mutation, and delta is of course the most dangerous one now. So mutation happens in the host body when it is multiplying.
Yes.
Is that so?
See, it is the virus. Once the virus comes on the mammalian cell, it injects its nucleic acid inside the mammalian cell so that the nucleic acid can get converted to protein. So the host translation machinery is used to convert this nucleic acid information into protein. So it is during this step that the mutation takes place. And since the virus is, it grows very fast in the sense that, in 12 hours or maybe 6 hours, they will have a generation. So the mutation can happen very often.
So it's basically happening in the host body, is it?
In the host body, yes, inside the host cell, because it is using, see, that is the typical of viruses. They use the host machinery to replicate its own nucleic acid, to replicate, and make the virions, the particles. So it is happening within the cell, and it is using the host machinery to do this.
Just last question before I close. If two host bodies are similar, will the mutation also will be similar?
I mean, similar term. The term.
Or basically what I was asking is that, is it the host body also playing some role in the mutation of the virus?
I do not think so. I think it's the machinery which is at the point when it's happening. It's the error. See, during the transcription, when it is happening, when you're forming here, so you have got the RNA already. So you're translating the RNA into the protein. And when the translation machinery is functional, there the substitutions take place. And so I'm not sure. By chance, see, in three of the variants, there is one amino acid which is very prone to mutation. So it can happen that the same mutation is occurring in the two bodies, or must have occurred like that, because in the variants that we get, there are similar mutations. So it's entirely possible.
Thank you.
Yeah. So I request the distinguished audience who have joined through WebEx to ask questions if they have any.
I can see the hand of Janardhan there.
Yeah, Professor Janardhan, yes.
Yeah, I'm Professor Janardhan from PRL. Thank you very much for that very informative talk. I am a space physicist, and so I have a very naive question to ask. Forgive me for that. You see, when the virus, this current COVID pandemic first started, they said it came through a wet market in China. And that is kind of logically understandable because people interact with the animals, they ingest the virus and all the things, and it has a long time to, jump from animal to human. My question is, can we now have possibly an engineered virus? Now, can these viruses interact with each other and pass on this engineering to something else? Then we'd be in very deep trouble.
Well, that's first suspicion, initial suspicion.
For example,
initial suspicion was that two viruses were infecting a same bat, and where their genes got exchanged. And so one virus became very infective. So this exchange of genetic material is very much possible. So when you get a more infectious virus, that genetic material can go into another one. If you are
Can it happen in
doing in the same, if you are infecting the same body. Excuse me?
No, can this happen in the
I can't
atmosphere,
hear you.
for
You must be muted.
example, in aerosols? Can this interaction happen in the aerosol particles?
No, it cannot happen because the virus survives only within the mammalian cells and functional. They can be only suspended in the aerosol, and there, I don't think it can happen there. No.
Yes.
Thank you very much.
Professor Palamraju?
Yes. Thank you. Excellent talk, madam. Very illuminating talk. I have a couple of questions. One is, do you think or foresee that this vaccines will become kind of flu vaccines that periodically we may have to take down the line?
Looking at the spread of the disease and looking at the persistence of it, I think it will become like that, that every year we may have to take a shot to, sort of survive this. And depending on how virulent or how infectious the virus gets with time.
Okay, thanks. The second question was, is the virus transmitting virulent more in dead bodies? Because when there were unfortunate deaths, it was in the news that they are more virulent there. How it was very difficult for me to understand.
No, they're saying more virulent because the person died. That's why they're saying, it's more virulent. But the most of these effects are due to the virus causing physiological changes within the body. So once it goes into the lungs, and then the lung has granulomas and it's causing the lungs to malfunction. That is you will notice that initially there were a lot of deaths, because at that point of time, either we were caught unawares or the virus was, changing at that time. So it's not that dead bodies have more infectious viruses. It's because the person died, they're saying that it has more infectious viruses, same virus that's affecting people. To some people will die, some people will not die.
Yes. So. Yeah, that's all. Okay, thank you very much. Okay, we'll let.
So we have another question that I'm reading from YouTube, and we have time. So vaccine is mRNA based. So does it interrupt normal physiological protein manufacturing and interfere with growth and development of young children?
No, it doesn't. So far, we have no evidence of that. When a virus infects a cell, when the viral RNA goes into a cell, what virus has a mechanism of stopping the host's own mRNA to be used. So that is when a virus does it. But in case of vaccines, when the liposomal RNA goes there, there is no other viral proteins that are actually can cause that kind of issue. So there is nothing like that, as far as I know. Now, what will happen after 10 years, it's difficult to say, because we have been there only 2 years, and in that 2 years we do not have enough data also to show that whether the mRNA vaccines can cause anything. It's a totally, see, the Covaxin or the other vaccines other than the nucleic acids are used technologies, proven technologies. But when it's nucleic acids, it's totally new technology, and it's being tested for the first time. So it's very difficult to say what are the effects that will happen.
Our doctor from PRL, Dr. Dhani, he has asked a question which I would like to read. Otherwise, he can also interact directly. Dr. Dhani, if you have a question, then you can ask directly. Otherwise, I'll read.
Yeah, thank you, madam.
Yeah.
I just want to, ask a single question. We have a beta, memory cells associated with our, beta cell lymphocytes. Then what is the role of, having this adaptive immunity against this COVID-19, particularly in our memory cell?
Yeah, you have to have memory cells. See, if you get a vaccine, there will be B cell memory generated. So the B cell memory remains in your lymph glands. So if you get an infection, then that B cell memory will be recalled, and then you will get enough antibodies to fight with the infection. So the memory B cells are actually required for, that happens like I said in case of chickenpox, like memory B cells are very strong. So memory B cells are there in your lymph glands, and when you are challenged with a say coronavirus infection, they will be recalled.
So, I mean, is there any association between the memory cell response and the, booster dose?
Yes, the booster dose actually the booster dose is responsible for calling the memory cells to generate antibodies. The booster dose is specifically to stimulate B cell memory.
Thank you.
Thank you, Dr. Darling. And we are almost done with, okay, there is one more question coming from YouTube. Among lot myth and facts about improving immunity to encounter COVID-19, what is your suggestion in Indian context? Useful things one should adopt as preventive measure to boost immunity?
I don't think one can boost immunity with any, with these supplements or anything. You have to have a very good diet to have a good immunity. And so there is, all these immune boosters that people are, giving advertisements, I don't think they work there like that. But in general, if it improves your general health, that is the best immunity you can have.
Thank you. There is another question: why the third wave is supposed to be the most dangerous for children when previous two waves had not affected them much?
Well, the third wave, they're saying that because the children are the only group which has not been vaccinated, so it will be very dangerous for them because the virus will not have enough to infect in the other population. So it may be very bad for the children. There are incidences of children getting the disease, but the reason for that may be because the rest of the population has been vaccinated surrounding that area where there was a report. But in India, there are a lot of people are not yet vaccinated. So I don't know how it will affect children selectively.
Yeah, thank you. So perhaps we have a last question from you.
One question, Lokesh, if there's time, can I ask?
Professor Deshpande. Yes, please go ahead.
Yeah. Good afternoon, and thank you very much, Professor Shaha, for such a wonderful talk. I have a couple of questions. First question was, you mentioned about 3 possibilities of vaccinating. So how is this vaccination approach of killed viruses? How does it generate the immune response? I mean, mild virus injection is okay, but how does killed virus injection generates immune response?
Yeah, so this killed virus is by fixing the virus with the formaldehyde. So when you fix something with formaldehyde or similar agent, what you do is you actually preserve the architecture of the surface proteins. So when that is preserved, then those epitopes are recognized by the macrophages, and those epitopes are the killed virus is chewed up and the antigens are presented. So the structure is maintained. So many of these are epitopes that are very hardy and maintained on the surface.
And in terms of developing the vaccine, this killed virus approach is more effective or more easier?
It's easier in the sense, I wouldn't say easier because you see, you have to grow the virus. So growing the virus is difficult. So you have to grow the virus and then fix it and then use it for vaccines. So it is actually not that easy, but when you are using nucleic acids, you can actually make them, in the laboratory. So you can make it in a large quantity. So killed virus in that way is a bit difficult because then you are, using a real virus from a culture. And you have to make sure that the virus are dead, and so they do random testing to see if that preparation is actually infectious or not. So that in a way is a little more difficult, I would say.
Right. And going by the analogy of survival of fittest, every successive mutated virus, will that be progressively stronger and stronger, which will be difficult for us to manage, or no?
No. The mutations happen, and mutations sometimes make the virus stronger, and they are very few. And there are mutations who are actually not of any use. So they are purged from the scenario. And the rest of the virus that has ability to infect more because of the mutation, they will be fitter and survive better. And so some mutations are, that's why the disease has spread like this, because some of the mutations have really become, it's giving more virus in the airways, so increased transmission. They bind more strongly, so that is also there. So their beneficial mutations are retained.
And the last question was related to your slide on the polio. That was an interesting observation that the polio cases started declining from 1982. So it was, your slide showed somewhere around 30,000 cases in 1982. And then it was declining. And in 87, once again, it shot to 28,000. So I'm just curious, was there a mutation in the polio virus, or that was just a relaxation in vaccination drive?
No, I think it was vaccination. It depends. The vaccination drive actually became slack. So, and when they gave the oral polio vaccines, then it became better.
Thank you. Thank you so much. Thank you so much.
Thank you, Professor Deshpande. And I request colleagues who have joined through WebEx, they can ask questions.
Lokesh, I have a question. Can
I
go?
Yeah, go ahead, ma'am. Yeah, please.
So my question was, with the experience that we have gained in the last 2 years or so, are there new efforts being taken to develop some kind of new tests or that can actually determine or predict the severity of infection in patients? Is there going to be a change in the direction of testing?
There are many investigations that are being done that how to predict the severity of the disease, how this X person will have a more severe disease, because it has everything to do with the immune system. So whether this person will get more severe disease and whether the other person will not get more severe disease. So diagnostic tests are being designed, but it's very difficult to do such kind of predictions, that whether it's going to be more or less. And one is to sequence the virus and see which one that you got infected with, and that tells you that whether you will get a more severe infection or not. But one cannot tell what your own immune system will do.
Okay. Yeah, thank you.
Yes. Someone had a question. Yes.
Lokesh, can I ask a question?
Sure.
Yeah, go ahead, please.
Yeah.
Professor
So, I'm reading it off from, this is Rubapati, from human to human transmission. Also, we are reading reports that there are human to animal transmission like cats or dogs. So is that really happening? Or
Yes, there are reports of cats and dogs harboring the virus and they are getting infected. So there are very few reports, but there are reports. So if cats can be, and they can be carriers also. So dogs, I am not sure, but cats have been shown to be carriers for this. And so if that happens, that really happens in large scale, then we are in really trouble, because we have, lot of us have pets of either dogs or cats, and if they become reservoirs for those that virus, then it will be very difficult to tackle. But there are evidences that has happened, not to a very large scale, but dogs and cats have been found to be carriers. Whether they actually get sick or not, I don't know, but they have been shown to be carriers.
So are there any other questions from our colleagues and the panel, people sitting in the panel?
Yeah, I would like to ask one question.
Yes, please go ahead.
Yeah, good evening, Professor Shaha. Thank you very much for such a nice talk. I just want to ask you one question, that ever since these different vaccines for COVID, they came into picture, one of the most effective vaccines they have been found out to be messenger RNA vaccines, but they need to be preserved in very low temperature. Is it because of the messenger RNA or the medium they are put in? It is because of what?
No, if it was medium, they would have rectified it. It's because it's messenger RNA. Messenger RNA is always very fragile to handle. So that's why it has to be kept in -80. But they have not yet found a way to stabilize it, outside that temperature.
Okay.
Oh.
Just one associated question. So far, most of the vaccines have been either dead vaccines or inactivated virus vaccines. There have been only some vaccines which use messenger RNA. Is it because of these problems with maintaining or sustaining the messenger RNA that the development of these vaccines have been a bit low?
No, these vaccines are a later concept. See, traditionally we have used the killed vaccine, and then generally the platform started to develop. One was DNA vaccine, and the vaccines. You see, the RNA vaccines and DNA vaccines came very fast. Why did they come fast? Because they were already being investigated for other diseases. But it was a completely untested technology which was allowed to be used, because you'll see that it is called that they are allowed on some. They have a clause. So these are actually, they came later. So that is why we don't have that much knowledge about them.
Okay, thank you very much, ma'am.
Okay. Lokesh, are there any other questions on YouTube?
Lokesh,
You are mute, Lokesh.
So there is more question in YouTube that is perhaps going to be the last question, I guess. So the question is the extent of side effects in response to vaccine correlated to the effectiveness of vaccine in that particular person?
Depends on what side effect it is. Actually, the clotting side effects that has happened, that would be in my, thoughts, they will be independent of the effect of the vaccine. Because if a clotting problem has occurred, it's due to effect on the different system, but its effect on the immune system will remain the way it is. So I don't think it will be.
Okay, thank you, madam.
Thank you very much.
Yeah, and taking a lot of questions from our audiences and very nicely interacting with them. And then I request our chair, Professor Nandita Srivastava, to take over the session for the further proceeding. Thank you.
Thank you, Dr. Lokesh and Dr. Maharaj, for conducting this question and answer session very well. Now I hand over to Professor Maharaj to thank the speaker and also the audience.
Thank you, Professor Nandita. It's always very difficult to end such a lively discussion, but due to the time limit, we need to do the needful. On behalf of the Director PRL, all members of PRL Ka Amrut Vyakhyaan, I would like to thank you, Professor Chandrima Shaha, for such an informative talk. I'm sure all of us are going to take back with us some enriched material on this topic of immunity and COVID. Thank you so much. I would also like to thank all INSA fellows for honoring us by joining us in this Amrut Vyakhyaan live today and participating in the discussions. Last but not the least, I would like to thank the audience who have joined us through YouTube link. Please do join us again next week, same time, for another interesting talk and round of discussion. Thank you all and have a good day.
Thank you. Thank you very much. Thank you very much.
Thank you so much, and please stay safe, stay healthy, and follow COVID appropriate behaviors.
Same wishes to PRL.